renal cell carcinoma, which accounts for 15 to 20% of renal cell carcinoma, is an heterogeneous disease that consists of various types of renal cancer, including tumors with indolent multifocal presentation and solitary tumors with an aggressive, high-level phenotype. Little is known about the genetic basis of sporadic papillary renal cell carcinoma and the effective forms of therapy for advanced disease.
These are kidneys. Each year, 65,000 people in the U.S. learn they have kidney cancer, also called renal cell carcinoma, or RCC. And 33% of those people will already have advanced disease at the time of diagnosis, meaning the cancer has spread beyond the kidneys to the other parts of the body. Understanding this disease will enable patients to take an active role in determining the best management plan. So let's get started. There are different molecular pathways that allow advanced RCC tumors to grow. The two known main ones being the vascular endothelial growth factor, or VEGF pathway, and the mammalian target of rapamycin, or mTOR pathway. The VEGF and mTOR pathways are active in the majority of RCC tumors. The VEGF pathway is primarily responsible for the blood supply, which delivers nutrients to the tumor. The mTOR pathway is primarily involved in the tumor's ability of cells to grow and multiply, as well as the blood supply and metabolism. The goal of treatment is to block the growth of the tumor for as long as possible. There are different options available, and doctors will often use several therapies, one after another, to try to slow and temporarily stop the tumor. Healthcare teams may use different types of treatments, including surgery, radiation, chemotherapy, cytokine therapy, and targeted therapy, depending upon a patient's disease.
Applications in medicine. Much of our knowledge of the genetic basis of papillary renal cell carcinoma has been based on the study of the inherited form of the disease. Hereditary papillary renal cell carcinoma, a rare disorder that is associated with an increased risk of type 1 disease, is characterized by activated germline mutation of MAT. Somatic MAT mutation occurs in the 13 to 15% of non-hereditary papillary renal cell carcinomas. Mutations in the genes that regulates the NRF2, ARE pathway, such as CUL3 and NFE2L2, which encodes NRF2, have also been observed in sporadic papillary renal cell carcinoma. Proof. They performed an integrative genomic analysis of 161 papillary renal cell carcinoma tumors to provide molecular insight into tumor classification, inform clinical recommendations, and suggest paths to the development of mechanistically based therapy. This classification of papillary renal cell carcinoma could potentially have a substantial effect on clinically, on the therapeutic management, and on the design of clinical trials.
So interesting in the context of kidney cancers that we have 7 FDA-approved drugs for the disease, but we actually don't apply any of these targeted drugs in a specific fashion based on a patient's genetic profile. So with the Foundation Medicine Company, I've actually been working with the group to profile a series of over 440 patients with advanced kidney cancer. I've done this in association with my colleagues at the Dana-Farber and at MD Anderson. But what we've yielded from this project is a very comprehensive analysis of this patient population and found multiple mutations that were previously undescribed in the disease. So with this project, I think we'll move one step towards actually identifying the right drug for the right patient. It could potentially lead to major breakthroughs. And I think that's going to be a big paradigm shift in the way that we do clinical trials in the future for this disease. So, in the context of this project, amongst these 443 patients, we actually have a smattering of individuals with rare subtypes of kidney cancer. Papillary kidney cancer is certainly one of them. At the U.S. Congress of Pathologists, we actually reported some data in that subset. We actually are reporting out data at this meeting pertaining to collected duct kidney cancer, and this represents just about 1% of all patients with advanced kidney cancer. happens to represent the most aggressive subtype of the disease. It's invariably fatal. It responds poorly to chemotherapy. The chemotherapy represents the standard. What we've uncovered in collecting duct carcinoma are mutations, several that may potentially predispose to certain targeted therapies. So, for instance, we've uncovered mutations in a gene called NF2. This may potentially lead to sensitivity to drugs like everolimus or Afinitor. So I can really see a continuum with this research that leads to targeted therapies for these rare subtypes of the disease.
This study suggests that gene fusions involving TFI3 or TFIB are underappreciated in type 2 tumors in adults and should be considered in any patient with type 2 disease. Although papillary renal carcinoma with fusions involving TFI3 or TFIB are generally considered to be disease of children and young adults.
For papillary cancer, for renal cell carcinoma, so there are two types. One is papillary type 1, the other one is called type 2. So type 1 actually has a higher frequency of the gene mutation. It's called MET mutation. So Dr. Schreier actually did a clinical trial with a MET inhibitor that showed some modest activity, but that c-MET inhibitor, according to this talk today, it's not a very pure inhibitor. So right now, I think they are planning to do another more pure MET inhibitor, and then hopefully, this new MET inhibitor will have more clinical activity in this type of cancer. So right now, we are still relying on drugs that are for the common renal cell carcinoma, except these rare types of tumor. But clearly, they have different drivers of gene mutation for this rare type of kidney cancer. So by using one class drug on another class or other classes of tumors, that may work well. So I think as long as we track the biology of this rare kidney cancer and design drugs to target these changes, the hope will be higher in the future.
Conclusion. Type 1 or type 2 papillary renal cell carcinoma were shown to be clinically and biologically distinct. Alterations in the NAT pathway were associated with type 1, and activation of the NRF2-ARE pathway was associated with type 2. CDKN2A loss and CIMP in the type 2 conveyed poor prognosis. Furthermore, type 2 papillary renal cell carcinoma consists of the least three subtypes based on molecular and phenotype.
Better understand papillary RCC, what you have to understand is the big picture. And so overall, out of 100 renal cell carcinoma, 80 will be clear cell. We're not talking about this one, we're talking about the 20 left. And 15 of these 20 left will likely to be what we call papillary renal cell carcinoma. And it's based on the morphology, how it looks like when you look at the tumor with a microscope. And within this family of papillary renal cell carcinoma, there are usually two entities that are what we call the type 1, which tend to be rather smaller tumors, more localized to the kidney, or what we call the type 2, that can evolve more rapidly to metastatic disease. So these are the kind of renal carcinoma. There is no specific risk factor for this subtype, so we stay on the field of age 60 with known risk factors as hypertension or obesity, overweight, are some inherited syndroma that does exist, and we can have that with papillary renal cell carcinoma. So there is some inherited syndroma with that kind of tumor. But what we do know is that for the first part of this papillary renal cell carcinoma, which are the type 1s, it's very likely that MET is a gene that codes for a protein that is on the membrane of tumor cells. Is very important because we know that it can be mutated, and so it means activated. And MET appears as a relevant biological parameters in that kind of type 1 renal cell carcinoma, papillary renal cell carcinoma, and it may play a role as well in few type 2. PRCC, as all non-clear cells, has no specific therapy available. We use by analogy with what we do with clear cell renal cell carcinoma, and the guidelines always state that patients should be referred to clinical trial so that we try to better investigate the disease and offer new therapy and all what the clinical trial brings to patients in terms of accessing new compounds, because the results with the available agents that are approved are quite disappointing so far.